I started my medical career as a resident in Germany. I was responsible for the lung cancer patients. Often cancer is considered as a homogeneous mass, but it's a very complex construct of cancer cells, and they can only survive in the context of normal cells, and therefore I believe that the truth is in the tissue. If you look in the microscope, you will see the cancer cells that are embedded in different tissue construct structures, and these are immune cells, these are fibroblasts, these are vessels, and they are all essential that the tumor can grow, and they interact with each other. These I often call a map of cancer. A map will help us to understand these interactions, and I think a map is the first step.
To make a map of cancer, the first step is immunolabeling. Imagine you're looking at a detailed black and white photo of a busy city. You can see every street and building, but you can't tell which ones are which. You want to find all the schools in this city, but from above they look just like everything else. Now imagine you have a kind of magic paint that only sticks to schools and makes them glow. Suddenly, the city starts to make sense. You can see where the schools are, how many there are, and how they're arranged across the city. That's what immunolabeling does inside the body. The schools are proteins, the small but vital molecules that help our cells function. The magic paint is made of antibodies, special molecules that recognize and bind only to a specific protein. When the antibody finds its matching protein, it leaves behind a colored signal. Under the microscope, those colors reveal where the proteins are located, creating the first layer of our cancer map, showing the tissue structure and landmarks.
My research group, both the research group here in Uppsala as well as the team in Bergen, at Bergen University, we are investigating how very early lesions of tumors, so at this stage they are still pre-malignant, they are not yet a cancer, but how they progress towards invasive cancer. And Patrick had a really good access established here at Uppsala to tissue collections. So this was a great opportunity for us to join forces and apply advanced spatial techniques to the valuable tissue collections that he has.
One major question in our research is, of course, to explain why some patients have a very good benefit of these new immunotherapy options or some patients have no benefit. And therefore we believe that it's not enough to look just on the proteins. It's not enough to look just on genes. And we believe we have another level, and this level, what we believe is a level of functionality. And this functionality is based on that we maybe can detect interactions and interactions that are relevant.
A city is not only defined by its buildings. What makes it truly alive are the connections, the power lines, the conversations, the shared spaces. Inside our tissues, the same thing happens. Proteins don't work in isolation. They interact, communicate, and sometimes form powerful partnerships. To reveal these hidden relationships, scientists use another method called proximity ligation assay, or PLA. If immunolabeling shows us where the buildings are, PLA shows us who's connected to whom. In PLA, scientists look for moments when two proteins are very close together, close enough to interact or cooperate. When this happens, a special signal is formed, a tiny point of light that appears exactly where those two proteins meet. Under the microscope, each light represents a connection, a conversation between two proteins.
So if two protein interact, this could indicate that a certain signaling pathway is actively signaling in the cells. So it provides the cells with some information that they should proliferate, migrate. So we want to inhibit these processes, and therefore we need to know the pathways that are active. So we started to use this assay in order to map the interaction of immune checkpoint proteins in the tissue context, because these are the proteins that are targeted with immunotherapy nowadays. So it was very valuable for us to know if these immune checkpoint pathways are actually active and thus can be targeted. So we see two ways how our research can benefit the patients. And one way is that we can identify new therapeutical targets that can be used to improve treatment options for patients. But on the other hand, we also believe that these spatial maps that we generate of the tumor will provide us with some markers that can actually predict if a patient is at a high risk to metastasize or to get a relapse, or if this patient has a high chance to benefit from a certain treatment.
Me as pathologist, I want to increase the possibilities to get the right treatment for the right patients, what we call precision medicine, that we have the best diagnostic tools to detect the best treatment for each patient. Our research confirms some kind of concept that we have, that immune cells interact with cancer cells and that they are responsible, that they can be used as a mediator for therapy. This is, I think, the future of therapy, that we use this own patients' weapons, body's weapons, to treat cancer.